Our final talk is going to be given by Dr. Ken. Your name isn't on your abstract.
Oh, Matt Kennedy. Yeah.
It's Matt Kelly.
Kennedy.
Matt Kennedy.
Dr. Kelly.
Ken Matady. Dr. Ken Matady, who is a postdoc with Brian Cullen. He got his PhD at the University of Rochester studying HIV reverse transcriptase. And he is the first representative talk for the new Duke CFAR subsection of HIV cure. He's going to be telling us about exploiting the CRISPR-Cas9 system to either excise integrated viral genomes from HIV-infected cells or possibly eliminating those cells, and also going to tell us, I think, about application of this system to HPV as well.
Viral malignancies.
Matt?
Thank you. Okay. All right. Thank you. If you guys can't hear me, just wave at me and wave me back to the mic. I tend to walk around. Thanks for the opportunity to speak to you today. Today I'm going to tell you about a good amount of work that a team of us have been doing to target viruses integrated in the human genome. So first, let me introduce CRISPR-Cas9 systems. And CRISPR-Cas9 systems are a fascinating example of adaptive immunity in prokaryotes and archaea. So for you immunology geeks, that may be especially relevant. But CRISPR-Cas9 systems occur in the host bacterial cell when a phage enters the cell, and then the phage is then chopped up and integrated into the host prokaryote archaeal genome. Now, today I'm going to talk principally about type 2 CRISPRs, of which this enzyme from strep pyogenes is a prototypical example, of which most of the work you see being done currently is being done primarily because there's a single effector. This effector can then be vectorized and be used in a bunch of molecular applications. So this effector is a double-stranded endonuclease that's guided by an RNA, or a series of RNAs. And basically, this nuclease has two central requirements for cleavage. They have trouble. You guys can't see this? Okay, great. A pair of RNAs that are encoded by the bacteria from repeats of integrated phage, of which one is called the CRISPR RNA. One part of that is part of the invader, which is then complementary to the target. So basically, that has a repeat attached with that, which then is annealed to what's called a tracer RNA. This pair of RNAs, in conjunction with a part of the enzyme that recognizes the other strand of DNA, independent of this cognate interaction, this piece is called the PAM. Now, the PAM is required for every initiation of cleavage by a CRISPR-Cas9 system. And the PAM exists on the other strand from the strand that the guide RNA is complementary to. So in the case of strep pyogenes, represented here in this lovely crystal, you can see that the PAM is a pair of Gs, and that those are recognized by a pair of arginines in the enzyme PAM recognition domain. So pretty much the PAM is recognized first by the enzyme, and then these pair of RNAs are fed in, and then cleavage initiates. So important innovations earlier, about 2 years ago, fused these two RNAs with a direct GAA tetraloop right here from 3' to 5', making this one RNA transcribable from a single pol 3 locus. So basically you can express this from a pol 3 promoter, you can express the enzyme from a classic pol 2 promoter, and then you have a functional Cas9. So I'm going to tell you about two small vignettes today. I'm going to highlight some data from some work we've recently published with integrated human papillomavirus. And HPVs, specifically serotypes 16 and 18, are causative of invasive cervical cancer, head and neck cancer, and a lot of people are infected. And even though there's a very, very effective vaccine, adherence is low. People really think of the vaccine as more of a sex-associated vaccine when it should really be considered a cancer-associated vaccine. But that's political commentary for another time. But if people don't adhere to the vaccine or are already infected and the vaccine is not known to be prophylactic, then you have a high incidence of head and neck cancer, which is devastating for those that get it in the neck because it has to be surgically removed. And then invasive cervical cancer, despite the availability of these vaccines for years, is still on the rise. So this is a very important viral malignancy. And similar to this, where the virus integrates, I don't need to go into depth here for this crowd, but the real problem here is work shown by many in the latency field that indicates that if people are desuppressed from heart, that is, heart is withdrawn, viral rebound occurs. And work from Bob Silicano and others has shown that the half-life of virus in the infected individual is practically infinite. So there's a serious push now to try and eliminate provirus from transcription latent compartments, especially CD4 resting memory T cells and less so macrophages, but these also exist in a number of compartments. So first I'm going to tell you a little bit about HPV-18, for which there's a lovely model I'm sure you've all heard of, which is HeLa cells. So when HPV-18 is in the epithelium, it exists principally vegetatively in a double-stranded DNA episome. But occasionally, aberrantly, that episome is integrated. And in that context, usually the master regulator is disrupted, and then E6 and E7, which I'll discuss in a little bit, are dysregulated and constitutively expressed. When these are constitutively expressed amongst other roles not depicted here, principally E6 downregulates P53, which is a classic protector of the genome, and E7 binds PRB and promotes its degradation, but it also liberates it from E2F, which then turns on cell proliferation. So these are classic viral oncogenes. So again, these are the real problem for invasive cervical cancer when they're integrated and then dysregulated. So the real question is, can we take these RNA-guided nucleases and then target these genes and show phenotype? So the first thing, as molecular biologists, we do is we want to make sure this technology works. So we use a fusion protein indicator assay, which takes a little bit of rev because we like it, and then we have tons of antibody for it, and then an in-frame target for these trippers, and then a GFP cassette or an F-loop cassette. And then we can target them in the steady state by co-transfection and look at knockdown by either frequency or MFI, and you can see that these guides knock down GFP in the bulk of wells, and then those residual cells, the MFI is greatly decreased. So specifically, you can also see that the steady state of the fusion protein is reduced by Western blot, which is really amazing because you see that in the presence of both of these guides here, or both of these guides here blotting for rev, that the fusion protein almost goes away. So these are highly effective guides, and we picked one effective guide for E6 and E7. So next thing we want to do is verify these function in HeLas themselves. So we can do what's called a surveyor assay. The surveyor assay detects indels introduced after repair of those cleavage lesions. And pretty much you denature the whole population of genomic DNA and then re-anneal it after a PCR DNA, and then the surveyor nuclease detects the bubble where a wild-type strand would be annealed to a mutant strand. And you design those primers so they should be exactly the right size, and what we see is highly specific surveyor activity, maximal by 48 hours for both of these guide RNAs, and then we wanted to shallow sequence these loci and make sure that we see CRISPR mutagenesis there and that this wasn't at all off target. And what you can see for E6, now this is the guide RNA I talked about earlier, and then here's the PAM. So you can see that we see a number of deletions, but the principal lesion recovered was an indel of an A, and that's the bulk of mutagenesis, and this has been reported by the Zhang group and by Eric Lander. And then you can see for E7, similarly, the highest lesion is a T insertion. And basically, this tells us that, yeah, we have it transfected, it's functional, and that we're getting very effective mutagenesis of these genes. So the next thing we wanted to do is try and recapitulate previously shown phenotypes in either CHA cells. I'm not going to show you that data today, but I'll show you some HeLa data. CHAs are HPV-16 positive. So basically, if you disrupt E6, then we should see P53 go up because it's not pushed towards degradation. And E7 also we should see RB go up, and then we should see downstream effectors of P53 go up, for example, P21. So if you look by Western blot, you can see when we compare a non-specific sgRNA, this is an sgRNA that targets a sequence that's non-HPV, or E6 or E7. What we see is when we look at E6, we see a substantial increase in P53 and its downstream effector P21. And then if you look at the E7 guide there, that we don't see any increase there, suggesting it's not off-target DNA cleavage. We have some transcomplementation data I don't have time for, but we've looked at this rather rigorously. And then you can also see for RB that we see an increase with E7, and remember that E7 has two roles, so only part of its role, two among many, is to degrade RB. So now we know in the cellular ensemble that we have recapitulated previous phenotypes, principally upregulation of P53 and RB in the case of their viral oncogenes. So the next thing we wanted to do was look at a competition assay. So if we put co-transfect these effectors and then GFP and then track these with flow, do these populations crash relative to the ensemble? What we see is a non-specific guide. The GFP population grows very well, almost to confluency, but the E7 and E6 populations drop out. So then we wanted to look at some cell cycle analysis, and this work was done by Bridget and Anand in the lab. And pretty much they co-transfected E6 and E7 with eGFP. And what you can see is we see classic cell cycle arrest with both effectors. And this has been shown previously by a number of groups, principally the DeMaio lab first. So the next thing is we wanted to do some classic growth curves, but the real problem with HeLa, that no one tells you, is they're very difficult to transfect. They transfect maybe 20 to 30% with nice reagents like Fugene. So we made some CRISPR encoding lenses that are specific and used very high MOI to get the bulk of the HeLa ensemble. And then we decided to plate these and do a traditional growth curve. So this is about MOI of 2, titered off the green control virus vector, rather. And what you can see is that mock transduced cells control LCE virus to make sure transduction efficiency is very high, and a non-specific CRISPR Cas9 virus. These guys are all tracking well and piggyback, where the E6 and E7 both drop out of the growth curve, suggesting that they're not growing and possibly eventually apoptosing. So a collaborator of ours in the Cas9 lab, Michael Goldstein, then wanted to do a survival assay. So what he did was he transduced these with a massive MOI around 37 and 40. So this is synchronous infection. And what you can see is you'll see a little bit of non-specific toxicity due to lenti, but we see that the bulk of these cells are dead by day 10. So we're very happy with this data. Basically, cleavage of the integrated E6 and E7 loci recapitulates classic genetic data, and this is proof of concept that SPY-Cas9 can degrade these genes, and then these cells are killed. So, I mean, these approaches can be used for a number of viral malignancies, head and neck cancer, cervical cancer, anal cancer, and EBV, nasopharyngeal carcinoma. So the next question is, does this work in animals? And a big push in the lab is to optimize these guys to be small enough to fit in AAV so we can do some animal work, because the pyogenase protein is way too big. So next I'm going to tell you about the HIV story, which you probably were waiting for throughout that one. But first, I'd just like to say that a lot of this data was done by, a lot of this data was obtained by a gifted young grad student lab named Adam Mefford. He's gifted in a lot of ways. But he did a great job on this. So basically what he did was he targeted HIV-1 TAR with 3 guides. And we are a TAR-TAT lab, so these are very highly conserved targets. The TAR especially so. Its disruption usually kills virus, and the most of isolates obtained have broken TARs because the virus is dead. So basically the TAR was targeted, and the interesting thing about the TAR for the HIV virologist is that cleavage of the TAR results in possible excision of the entire provirus, which is very interesting. There's 2 targets after reverse transcription. And then the TAT gene is also very important for progression to elongation during HIV transcription. So, I mean, if we mutagenize TAR or knock out its target, then we should be able to try and eliminate the virus. So again, we screened a number of guides by a GFP assay. The TAR guides and the TAT guides are all highly effective by Western and by a GFP knockdown assay. And next, we wanted to ask the simple question, are these nucleases, when loaded with cognate HIV guides, protective against HIV infection if they're delivered beforehand? So we can make some target cells by putting in receptors with 293T, so CD4 and CD4 and CXCR5 should be, or 4, CXCR4. And then we can put in our cognate SPY Cas9 guide RNAs, wait a while for all these vectors to be expressed as well as receptors, and then infect with the NL4 loop virus that has loop in the place of NEP, and then assay for luciferase. And what you can see is that for the TAR guides, there's little or no luciferase produced across the board for these guide RNAs, which is somewhat amazing data, and I'll show a lot of data to support this, but we were very impressed. And for TAT, we also see a very impressive knockdown against incoming HIV in these cells after they're expressed. This is not super surprising, but it's also very interesting because a lot of the post-reverse transcription products are thought to be very heavily shielded by protein, well protected before integration. So the next thing is, if we take luciferase out of there and repeat that same assay with an actual infectious clone, infect those cells, and then look at virus coming off those cells to see its specific infectivity, and then titering on TZMBLs, which were discussed briefly earlier, but TZMBLs have an LTR driving luciferase. So basically, if you have infectious virus, then TZMBL produce luciferase. So what you can see is relative to a non-specific guide, all the TAR guides were very highly functional. And they're about at the level of background for naive TZMBL. So this data is very promising, and looking forward to following up on it. But the real question here is there's two possibilities, right? You could have the virus proceeding through reverse transcription, then nuclear import, then it could be integrated, and then the provirus could be excised. Or the proviral genome is cleaved before integration, right, and then degraded in the nucleus. So the real way to test this is to infect cells and then give TAT back with TAT guides. And I'll show that in a second. So basically, if we hit TAT and get rid of it, get rid of most of it, and then add exogenous TAT back, if there are proviruses integrated that just have broken TAT, then we should see a rescue, right, with exogenous TAT. And what we see is that's not the case. So when you add TAT back, plus or minus, you don't see any rescue of the phenotype, which makes us think that these SPY Cas9 sgRNAs are cleaving up the pre-integration complex, which is very interesting because it's thought to be very protected after reverse transcription. So the next question is, let's do the contrary. Can we remove latent provirus, integrated provirus from cells that are already infected? So we use the JLAT model here, and JLATs are a JERCAT line that are infected with a GFP reporter virus, sorted, activated, then individually cloned. And principally, they're dark. They're GFP negative until they're hit with a formable ester like PMA, which is usually what's made to activate T cells in the primary case. So what we wanted to do here is we made a lentiviral vector that encodes Cas9, and this was actually not trivial because the guide RNAs themselves hit the packaging vector, which for the HIV people, they'll understand that. But we made it, and then we transduced those cells and selected with puromycin. And then we added plus or minus PMA after time, and then we looked either for GFP positivity or with flow, or looked for P24 with Western. P24 is the capsid protein, as you guys know. So what you can see is that with both TAR guides, we see a drastic decrease in GFP after PMA stimulation. And then if we look at P24 production for the non-specific guide, which doesn't hit anything in either of the TAR guides, either plus or minus PMA, this is the classic way to look at HIV production, the non-specific does nothing off target. We see tons of P24 produced. And then we do see a little residual P24 here, almost at the level of background. But this is known with PMA because it's a very strong stimulator of NF-kappa B. NF-kappa B is all over the LTR. So this is expected with PMA alone without a CRISPR. So with that, we've shown that both before infection and after infection, we can disrupt HIV, especially with the TAR and the TAT gene. And the fact that Cas9 is capable of preventing infection sort of goes against the reverse transcription dogma that the pre-integration complex is well protected before integration. So that's very interesting. And then finally, we know that we can possibly move to some better models of latent infection in chronic cells. So with that, I would like to first thank two very good trainees. One is Adam, who did a lot of the HIV work here, and then another one is Anand, who has worked with me for a long time and has helped with the HPV work. I'd also like to thank the rest of the people I work with regularly in the lab, Hal, Adam, Bridget, and Joy, and then Michael for the cell survival assay, and then Duke and Roche for the money and support. So thank you for your time.
Any questions for that? Comment and question. I mean, one way to directly prove particularly the pre-integration effect is using integrative effect of the virus.
Or user integrity.
Right. Most of your assays don't absolutely prove that.
Oh, we haven't begun to prove it.
Yeah.
It's just it's like a good smelling pie in the window that we're trying to.
The other big question relative to safety is off-target effects.
Oh, with CRISPRs? I think that is a very important question, and there's a substantial controversy in the literature about that. But if you consider zinc fingers, for example, which are far advanced in clinical trials, their specificity is less in proportion. Right, so, and then the other thing to consider is
It's not
that...
about relative, it's about absolute safety, right? Because for this
No, I mean, it's in clinical trials. I mean, absolute safety is important for people that are maybe getting some gene therapy for something that's not essential, but for people that are terminally ill with cancer or people that are terminally ill with disease, obviously there's a therapeutic index, correct?
But HIV is not that disease.
Not now. But the important thing to remember with CRISPR, that's absolutely a good comment. But people bring that up with CRISPR, but then there's tons of lower metazoans, monkeys, cattle, mice. They've all been extensively manipulated without any serious evidence phenotypically in vivo. But I think it is valid concern. I mean, that's something for biotech to do, like very high fidelity CRISPR. That would be something I think the FDA would.
About the effectiveness of this strategy in red team cell. So I'm replicating CD4.
Oh, and say CD4, resting CD4 T cell. So you're more concerned that it wouldn't be expressed?
I don't know.
Well, I mean, there may be two issues there. One is delivery, which is the million-dollar question, right? Finding them and delivering it. The second question is, will they be effective against the targets in those cells? In contrast to talons and zinc fingers, CRISPRs are insensitive to methylation. So I think that's important. I mean, a lot of those loci are silenced.
Any other questions? All right. Thank you very much.